In the module you've just looked at, we looked at an overview of energetics in nature. In a class I prepared previously, Biology 101, we look at the energetics in more detail. So I would like you to watch this next supplement, and it will look at some components of energetics in a little bit more detail than what I just discussed. Today we're going to continue our discussion of energetics and what I call pieces of the energy puzzle. And so I'm going to be describing a number of different phenomena and processes that are important to the overall process of energetics. But we're going to be talking about them in isolation. So it's going to be a lot like looking at a little piece of a puzzle and trying to figure out what that piece is. And that's going to be difficult right now. You may not really understand what these processes are until we put them together. And these pieces will eventually form a puzzle, one being photosynthesis and one being the cellular oxidation of glucose. But we'll deal with those in future modules. So don't be too concerned if you are a little confused at this point, but hopefully by the end of photosynthesis and cellular oxidation of glucose, it crystallizes and makes sense. All right, so let's begin with oxidation. The term oxidation means a molecule or atom loses electrons. So if a molecule or atom loses electrons, that's oxidation. And also, it's sometimes hard to know whether electrons are being lost, so sometimes we track hydrogens instead of electrons. Now, reduction then would be the gain of electrons. So again, these are important terms. I'll be using them a lot, and sometimes students say, when I say something has been reduced, they'll say, well, why did it get smaller? This has nothing to do with big or small. This has to do with gaining electrons. So if something is reduced, it gains electrons or hydrogen atoms. So, for example, if we have substance X then that has an electron, it's going to give it up to substance Y. Substance X, when it gives away its electrons, is oxidized. Substance Y, when it picks up those electrons, becomes reduced. So we normally look at oxidation and reduction occurring together, because if I'm giving away electrons to something else, I'm becoming oxidized and it is becoming reduced. Even something like an ionic bond then involves oxidation and reduction. So you may recall that sodium will give up electrons to chlorine. So sodium gives its electrons to chlorine, so sodium is oxidized in that process. Chlorine is reduced in that process. We can also again look at hydrogen. So if we look at the carbon-hydrogen combination here, the carbons, we have carbon-hydrogen on this side, we have carbon-oxygen on this side. So the carbons then are being oxidized. Oxygen picks up hydrogens to form water, so oxygen is reduced in this particular reaction. Okay, now, this becomes important when we look at enzyme function. So there are molecules called coenzymes and other molecules called cofactors which work with enzymes. Just like a coworker works with you at work, coenzymes and cofactors work with enzymes to help them function. Now the difference is that a coenzyme is an organic molecule, a cofactor is an inorganic molecule. We are going to be interested in coenzymes. And in particular, we're going to be interested in coenzymes that function as electron carriers. So electron carriers transport electrons from one place to another, usually in the assist to some enzyme that's carrying out some function. We will be particularly interested in three coenzymes over the next couple of modules. One of these is called NAD, and NAD stands for nicotinamide adenine dinucleotide, and that's why we call it NAD. And this is the structure of NAD, just so you can see it's a very complex molecule. You don't really need to know that structure, but just give you a sense of the complexity of the molecule. And another will be NADP, nicotinamide adenine dinucleotide phosphate. And then a third electron carrier of importance to us is FAD, flavin adenine dinucleotide. So these molecules are derived from B vitamins. And so vitamins are something that we have to get in our diet. And a lot of times people will say, well, you need vitamins for energy. That's true. You don't need them for the caloric value, kilocalories that they have, but they do assist in the overall pathway of energetics. And again, particularly these B vitamins that we're going to be talking about. Okay, now, these are electron carriers. So NAD will bind 2 electrons and a hydrogen proton, and it will be reduced to NADH. So that's the reduced state. And notice that the arrows go in both directions. So NADH then can transport those electrons to another location and give up the hydrogen and electrons and be oxidized back to NAD. So this is an important concept to understand, that these electron carriers can be reused. They can transport electrons to one place and then give up those electrons in another place. NADP, again, would be another example. NADP can pick up 2 electrons and a hydrogen, and it's reduced to the molecule NADPH. NADPH then can release that hydrogen and those electrons to some other molecule or some other location, and it's oxidized back to NADP. And then last but not least, FAD can pick up 2 electrons and 2 hydrogens. It's reduced to FADH2. It can give up those hydrogens and electrons to another molecular location, and it's oxidized back to FAD. So let's go ahead and look at the role of electron carriers in something that we videotaped a little bit earlier. Okay, what I'd like to do is a little analogy looking at the role of electron carriers in the way that they work with enzymes to accomplish a function. And so the analogy here is that I'm a construction worker, and my job is to separate these 2 planks that are held together with screws, and I'll use a screwdriver to do that. And let's just say in construction it's always nice to have an extra set of hands, and so John is going to be my co-worker, and he's going to help me accomplish this task. And so the other side of that, though, on the molecular side, is that I will also be representing an enzyme that's going to separate these 2 molecules. The screws will represent electrons, and I'm going to need John to get those electrons out of here. So I need his help to dispose of the electrons. So I'd go ahead then and bind my planks and my tool, and I'm going to go ahead and remove one of the screws. So I need John's help then to take that electron and get it out of here. So when he binds that electron, he's in the reduced form. He'll take that electron out of here, dump it somewhere, that returns him to his oxidized state, and now he can help me out again. So in other words, I wouldn't be able to finish this job if he wasn't here. All right, so he's going to take that electron, he's in a reduced form, off he goes, he's going to dump that electron somewhere, and now he can be reused again. He's in the oxidized state. So again, I need his help to get rid of that electron. He's going to take that electron, get it out of here, and now that allows me to finish my job. I can get rid of my screwdriver and separate my planks, or separate the molecules in this situation. So again, as an enzyme then, I would require him to finish the job. If he can't get those electrons out of here, I can't separate the 2 molecules, I can't separate the planks, I can't finish my job. So some enzymes then absolutely require coenzymes, in this case electron carriers, to help them finish the job. Okay, another concept we need to consider is that of an enzymatic pathway. Now, an enzymatic pathway, when we look at chemical reactions, as I've mentioned before, enzymes are very specific in what they do. So a lot of times a number of enzymes have to work together to accomplish some major task. And so when you start with a certain molecule and an enzyme works on it and alters it in some way, and then a second enzyme works on it and alters it in some way, and a third enzyme, and so on, and you eventually end up with a product that is distinctly different from your starting molecule, we call that a pathway. So you're going to start at one location, you're going to end up at another location in a pathway. Here we're going to start with one substance. We're going to end up with a different substance at the end of the pathway. Let's take, again, a fairly low-tech example using our overhead camera then of a chemical or enzymatic pathway. So let's say then that there is some molecule comes into the cell, and an enzyme will recognize that and say, oh yeah, my job is to go ahead and break that in 2. Now, another enzyme will say, okay, now I recognize this, I'll go ahead and break that in 2. Still a different enzyme will say, oh, I recognize this molecule, my job is to break that in 2. Still a different enzyme will recognize this molecule, bind it, and say, hey, my job is to break those apart. Still a different enzyme might recognize this molecule and say, hey, my job is to break those apart. Still a different enzyme might recognize these and say, my job is to separate those. But the point is that there are a number of different enzymes at work here, and what we end up with then is something completely different than what we started with. So when that happens, we have an enzymatic pathway. All right, let's contrast that then with an enzymatic cycle. In an enzymatic cycle, you start and end with the same molecule. and you bring materials in and you take materials out. So let's take, again, a kind of an analogy, low-tech analogy here. Let's say that I'm going to be representing both an enzymatic cycle and also we're going to be looking at a manufacturing process here. So let's say that I work in a factory in the days where there actually were workers and actually in the days when robots didn't do everything. And let's say that my job is to make 3 packs of water. So in manufacturing, a lot of times you'd use a mold, something to hold the parts while you constructed them. And so you'd bring in materials, take out your product, but the mold was always there. So in this case, we're just going to use this baking pan as the mold. And so let's say then that I'm the first worker. My job is to go ahead and put a water bottle in the mold. And then I pass that off to another worker, which would be a different enzyme. And so that enzyme or worker would take that, add another water bottle or add another part to that molecule, and then pass that off to still another enzyme or another worker, and that worker would go ahead and grab still another bottle and put it in the mold. And then that would be passed off to still another worker who would bind those together. And this wouldn't be a very attractive product, I wouldn't think. And then maybe that's passed off to another worker to try to finish that process. And so at the end of the process then, you would have your 3 packs of water, which would be removed, but you'd be left with your mold that you started with, in this case our baking pan. So the point is, a number of different enzymes would be involved. Materials come in, products go out, but you start and end with exactly the same thing. So that's the concept behind an enzymatic cycle. Let's use our overhead camera again to look at an example of this as well. So let's say that there's a molecule that we're starting with. And let's say a new substance comes into the cell. And so, again, I'd be an enzyme that says, oh, hey, this is supposed to be attached to this, and I might do that job. The second enzyme then would recognize this molecule and say, oh, hey, this piece is supposed to be attached to that. That's my job, I better do that. Still another enzyme might come along and say, hey, those 2 things aren't supposed to be attached to that, I'm going to break those off. So at the end of the process then, it's involved a number of different enzymes. We've had individual pieces come in. Those pieces are bound together going out, but we're going to start and end with exactly the same molecule that we started with. So again, the idea behind an enzymatic cycle is that certain particles come in, a product goes out, but you're left with what you started with. Okay, next, a substrate level phosphorylation. A substrate level phosphorylation is the simplest type of phosphorylation, and a little bit earlier we actually did a little videotape to show an example of a substrate level phosphorylation, so let's look at that now. Okay, next I'd like to do a demonstration of a substrate level phosphorylation. And so Justin is going to represent a molecule that has a phosphate, and so the tennis balls are going to represent a phosphate. And John is going to represent an ADP. So remember, ADP has 2 phosphates. So what I would do in this role in a substrate level phosphorylation is that I would be an enzyme that would bind a specific substrate, in this case Justin, and then I would bind an ADP, and that would cause a change in my shape to put them together in a way that they would transfer, that Justin would transfer his phosphate to John. So once he picks up that phosphate now, he's no longer ADP, he's ATP. Very important molecule. I'll release him, I'll release Justin, and now I go back to my original shape and I can bind a new Justin and a new ADP, put them together to produce more ATPs. So again, a substrate level phosphorylation then just involves an enzyme that causes a transfer of phosphate from one molecule to an ADP. It's the simplest way to make ATP. It's also the rarest way to make ATP. So just diagrammatically looking at a substrate level phosphorylation again, so what we would have is an enzyme. It will bind some substance that contains a phosphate. It will also bind the ADP, and it will transfer the phosphate to the ADP, releasing the original substrate now in a new form, and releasing ATP. So again, simple way to make ATP, but unfortunately a very rare way to make ATP. Just to give you a sense of how rare, if we start with a glucose, we can make about 36 ATPs from that glucose, and only 4 of those glucoses would be produced through a substrate level. Only 4 of those ATPs would be produced through a substrate level phosphorylation. Okay. Now, by far then, The majority of ATP molecules in nature are produced by another type of process, and we refer to that as an oxidative or chemiosmotic phosphorylation. And there are two components to an oxidative or chemiosmotic phosphorylation. One involves an electron transport system, and the other aspect of this is ATP synthase. So earlier, again, we taped an example of what an electron transport system is, and so we're going to start off kind of a low-tech example and work our way up to a more high-tech example of an electron transport system. So let's look at an electron transport system. Okay, one of the really important parts of oxidative phosphorylation is the role of an electron transport system. And so we're outside the studio today to give an example of that. So these three people are going to represent an electron transport chain or an electron transport system. And I want you to imagine that they're actually stuck in a membrane, so they can't move. And really their only role is to pass electrons. Now, if we're going to have electrons in, we need a source of electrons. So let's go ahead and introduce a source. So there has to be some molecule that's not stuck in the membrane that can bring electrons in. So let's have our source of electrons introduce some electrons, and members of the electron transport system will pass those down. So you can see we have flow of electrons. But once everybody has their complement of 2 electrons, and they're normally passed 2 at a time, the flow stops. So at this point now the electron transport system is not functioning. So in addition to a source of electrons that are free from the membrane that can bring in electrons, we also need a terminal electron acceptor, some other molecule to come in and pick up electrons and get them out of the system. Once those electrons are gone, now electrons can be passed through the electron transport system, and now we get a flow of electrons. So again, what's crucial to an electron transport system functioning, what we want is a flow of electrons. And if we're going to have a flow of electrons, these guys are stuck in the membrane. We need some molecules that are not stuck in the membrane to bring in electrons, a source of electrons, and then a terminal electron acceptor to take electrons out of the system. So again, as long as we have flow, this is a good thing. So a pending question is, well, what's the point? Why do we need flow of electrons? And so that's really the next step. What we're going to do is look at a more realistic example of an electron transport chain, and we're going to learn why it's so important to have flow of electrons. So bottom line then, electron transport system, we need a source of electrons on one end, we need a terminal electron acceptor at the other. That creates flow of electrons, which is a miniature electrical current. So again, what's the point? What is the point of having an electrical current? Well, again, that electrical current can do work. And the work we want it to do, now if we look at another example, is if you look at this diagram, then this line here and this line here are representing a plasma membrane. So you need to use your imagination and fill in the phospholipids here. And this is representing our electron transport system. And as electrons come into the system, flow through and come out of the system, what we want them to do is to pump quote-unquote hydrogens. We need them to move hydrogens from one side of the membrane to the other side of the membrane. So let's go ahead and look at how that is accomplished, because that again is the point of a flow of electrons. So the black dots are going to represent hydrogen protons. The white circle will represent an electron. So electrons are delivered by some kind of a source, and then the hydrogen protons will be attracted to the electron and they'll passively follow along. And so we're seeing the electron getting passed into what's known as a pump molecule. The electron moves across the membrane and it's going to drop the electron off on the other side. So again, the proton was taken in, moved across the membrane and left on the other side. And now that electron will continue through the electron transport system. Again, it will pick up another proton, move it across the membrane, dump it off, and eventually get picked up by the terminal electron acceptor. So at the end of this process then, as this electron is moved across the membrane and dumped the hydrogens on the other side, we refer to that as pumping. So what accomplished is that hydrogens are pumped across the membrane and concentrated on one side of the membrane compared to the other. So let's look at that one more time then. So again, we have protons on one side, and they're going to be brought in, moved across the membrane, dropped off on the other side. And then the terminal electron acceptor will pick those up. So as electrons move through, hydrogens are pumped. We end up with a high concentration of hydrogen ions on one side of the membrane compared to the other. So now the next question you might have in your mind then is, well, what's the point of that? Why do we want to pump hydrogens across the membrane? Why do we want to create a hydrogen ion concentration gradient? Well, these hydrogen ions now, of course, want to diffuse. They want to diffuse from where they are in higher concentration to a lower concentration. But they can't get back across the membrane. They can't get through the pump molecules. And as it turns out, there's just really one place where they can get back through the membrane, and it's a molecule known as ATP synthase. So again, this is a little more realistic example. Here's our electron transport system. As long as we have a source of electrons at one end, terminal electron acceptor at the other, electrons will flow through the system. As electrons flow through the system, they will pump hydrogens. Hydrogens are concentrated on the other side of the membrane. They try to diffuse back through the membrane, and the only place they can diffuse back through is this ATP synthase. So the hydrogen ions then are just following their concentration gradient. And as they move through the ATP synthase will have bound ADP and phosphate, and that will cause them this ATP synthase to change shape in a way such that the ADP and the phosphate are put together to generate ATP. So again, here's a close-up of the ATP synthase. Protons move through, ATP is generated. And so this is chemiosmotic phosphorylation, also known as oxidative phosphorylation. So let's look at a simple graphic for this, animation for this. Again, we have hydrogen protons concentrated on one side, and they're going to follow their concentration gradient. In the meantime, ATP synthase will have bound an ADP molecule and a phosphate. And so as the protons come through, usually two at a time, that will cause change in shape such that the phosphate is transferred to the ADP. That produces ATP. ATP is released by the ATP synthase. So again, the energy for this really is the kinetic energy of the protons. As they're moving through, those charged particles will cause a change in the shape such that ADP and phosphates are bound to one another. So let's look at, again, another quick animation of that. So the ATP synthase will bind ADP, bind phosphate, and then as the protons move through, it causes a change in shape and release of the ATP. Now, let's look at an animation prepared by our publisher, really, which will put all of these together. And we can see how these processes all work together. All right, so again, what we're looking at here, first of all, is the source of electrons. So the electrons are going to be the yellow circles. And they're delivered to an electron transport system and move through that system. So one of the things I want you to watch for then are the electrons being delivered. They'll move through the electron transport system, and then at the other end, they're going to be picked up by a terminal electron acceptor. So electrons are delivered, they're going to move through, and eventually be picked up here. Now the other thing to watch for are the little blue circles. Those are representing the hydrogen protons. And as electrons flow, hydrogens will be pumped across at several different locations and concentrated on this side of the membrane. So again, we can see protons moving through there, they're moving across here, they're moving through here, and they're moving through here, and concentrated on this side. So it's going to zoom in and show us the flow of electrons in a little more detail. You can see the protons also being carried. Again, the actual detail isn't that important as long as we understand, as long as there's a source and a terminal electron acceptor, electrons flow, hydrogens get pumped. And then that results in an intense concentration of hydrogen protons on the other side of the membrane. They want to diffuse back across the membrane. And of course, what's the only place they're going to be able to do that is through ATP synthase. So they simply follow their concentration gradient back through the ATP synthase. And again, this isn't a great example of showing this, but what's happening is the ATP synthase then is binding ADP and phosphate, and then the starbursts are representing the release of ATP. So the flow of protons in is causing the shape change responsible for the binding of the ADP and the phosphates. So we're investing a lot of energy, in a sense, in the flow of electrons in to pump hydrogens to generate ATPs. And ATPs then, of course, are a usable source of energy. Now, one more little puzzle piece I want to show you very quickly, and this is something I'll come back to again later, are photosystems. And where we find photosystems are in chloroplasts. And of course, if we look at a plant, the leaf has cells. The cells have chloroplasts. And you may recall that the chloroplast is a double membrane structure, and I'll talk more about this when we get to photosynthesis, and that inner membrane is highly folded. And so where we're going to find photosystems is embedded in the surface of these membranes here. These are known as thylakoid membranes. So we're interested in looking down on the surface of these thylakoid membranes and looking to see what kind of molecules we find there. And so what we find then are these photosystems. The yellow molecules here are representing pigments which absorb light energy. And then the green squares are representing chlorophyll. And chlorophyll is a really crucial molecule in the process of photosynthesis. So what a photosystem is all about then is gathering light energy. And what most of these photosystem molecules do is that they can absorb light of specific wavelengths. And when they absorb a photon of light of a particular wavelength, what it causes is the electron to go from its normal orbital to an orbital further away from the nucleus. So that takes energy. So the absorption of energy causes an excitation then of that molecule. Now it can't sustain the electron in that particular orbital, so it will move back to its original orbital. But then the pigment will pass that energy to an adjacent molecule, exciting it. So again, an excited state then is moving the electron to a more distant orbital. And then as the electron moves back, it releases that energy. So most molecules would release that energy actually as a burst of light and would be fluorescent. But this is what's known as resonance transfer. So as we look at our diagram here again then, what happens is that as these different pigments absorb light energy, they will be excited. And then they will pass that energy to adjacent molecules and excite them. And the real key is that many of these will focus that energy on chlorophyll A molecules, a specific pigment in these photosystems. And they will excite the chlorophyll A. So the chlorophyll A molecule then, if it's been excited by other photosystem molecules, and then if it gets struck by a photon of light, it will lose that electron. And it will eventually lose that electron to an electron transport system. So this is a really important aspect because once that electron is lost by chlorophyll A, that's the beginning of photosynthesis. So again, what we've done today is really look at little pieces of a puzzle. And my chore in the next couple of modules then are to take those pieces and put them together in the processes of photosynthesis and cellular oxidation of glucose.